Bioinfocongress IV. Assistant Professor Dr. Ahmet Coşkun. Secrets of Healthy Life Hidden in Cell and Tissue Architecture. Assistant Professor Dr. Ahmet Coşkun is currently a Burnham Marcus Early Career Professor of Biomedical Engineering at Georgia Institute of Technology and Emory University. Dr. Coşkun is a systems biotechnologist and bioengineer working at the nexus of multiplex cell imaging and quantitative tissue biology. Dr. Coşkun directs an interdisciplinary research team at the Single Cell Biotechnology Laboratory, an interdisciplinary program that is strategically positioned for multiparameter imaging one cell at a time by spatial context and function. Dr. Coşkun holds 5 issued patents and is also the co-author of more than 40 peer-reviewed publications in major scientific journals. Previously, Dr. Coşkun was an instructor at Stanford University. Dr. Coşkun received his postdoctoral training from the California Institute of Technology. He holds a PhD degree from the University of California, Los Angeles. His research has been supported by the National Institutes of Health, Wellcome Leap, Burroughs Wellcome Fund, NSF-CMAT, American Lung Association, American Cancer Society IRG, Multicellular Engineered Living Systems, and Regenerative Medicine Center. He is a recipient of the Student Recognition of Excellence in Teaching, Class of 1934, Course Instructor Opinion Survey Award. In addition, he leads outreach programs to engage K-12 students and undergraduate students through BioCrowd Studio, an innovative crowdsourcing program bringing together interactive virtual media distributed by kits and collaborative spatial discovery.
Dear Dr. Ahmet Coşkun and participants, welcome all of you to our Bioinfocongress IV event. Dr. Ahmet Coşkun will give us a talk today on Secrets of Healthy Life Hidden in Cell and Tissue Architecture. At the end of the presentation, we will have a question-answer section prepared by our team and consisting of questions from our participants. Dear Dr. Ahmet Coşkun, welcome back. We're so glad to have you here.
Thank you, Efekan, for the kind introduction and also for this invitation. And I'm very excited to be here and also share some of our work with the students in especially bioinformatics. And also thank you for this also wonderful organization. I think it will help increase the impact of the international science, right, through online forums like this one. And therefore, I appreciate your support and your efforts on this. So yeah, I'll get my...
Oh, sorry, sir. I'm sorry. Please.
I was just going to say I'll get started, but do you have anything to add?
Oh, no, please.
So then we'll get started. Thank you for sharing the slides. So today we're going to talk about architecture, right? So particularly if you look at this slide on the left, you're seeing the building that was designed in the shape of a cell and how also intercellular or multicellular structures would look, right? The architectures, they designed these buildings, and on the right, you're looking at multiple cells. Inside, you're looking at the specific organelles and nucleus in the purple color, right? And the organization also is there. So what's interesting is that there are similarities between a building and cells, right? So today I'd like to highlight this. So therefore, why cell and tissue architecture matters, right? Why is it important? For instance, when you look at inside a building or inside a flat or an apartment, you'll be looking at different compartmentalized parts, right? You'll be looking at the bathroom, you'll be looking at the kitchen and study room, right? And the living room and whatnot. When you focus and zoom into a cell, then you'll be looking at the organelles, right? The endoplasmic reticulum, the mitochondria, the nucleus, and whatnot. So I'd like to, give a general analogy between the two. First, parallelization. So in an apartment or house, actually you can do multiple events. While your family member cooks here in the kitchen, right? Then Some of the other family members, they can study in the study room, and some of the others can watch TV, right? So therefore, multiple events and processes can go in parallel. In the cell also, while some proteins are getting phosphorylated to perform a function, while the mitochondria give rise to more energy, some other organelles, they perform the waste disposal, right, of the cell, and some others also carry the proteins from some location to other locations. So therefore, a lot of parallel events. Second, the compartments. If you don't have a compartment in an apartment or in a cell, then it would be challenging to perform in a separate multiple events, right? For instance, the kitchen, the cooking will interfere with studying, right? Or the living room will get interfered with another thing happening in the laundry room, right? Same thing with the cell. If you didn't have a compartment such as mitochondria, all these molecules would not be dense enough to generate enough function to perform especially the necessary functions of the cell. Then it's goal-defined, meaning that if this cell is a kidney cell, then it performs the function of a kidney. If it is a cardio cell, then it performs the function of a heart. Then if it is a brain cell, then it performs the function of a brain. And similarly, an apartment or a house could be a personal use, or could be a business use, right? Or could be a study, a school or another facility, right? That can play the specific functions. So therefore, you can design it, and users also can design it. And the cells, of course, they self-assemble and design themselves, but they are modular. Then they're also tolerant, right? So in the case of failures, for instance, cells reassemble, change the positions, and change the particular number of organelles and their size and whatnot, right, and show us. So in the case of apartments as well, if you are not comfortable with certain passages, right, and how apartment is organized in terms of the locations, then you can reorganize to the best of your ability, of course. And therefore, there are a lot of things in common, right? And when you then look at the health and disease and zoom in to, for instance, outside the cell and inside the cell, then you're going to see that this architecture also changes between health and disease, right? For instance, the first right image, I will highlight this image. That's an organelle and 3D image of a subcellular region from a healthy donor. And the one on the right is from an obese donor, which means that that's not non-healthy, it's obese donor, right? So in the obese or non-healthy cases, then you start seeing these yellow circles popping up, right? These are the lipid droplets. So this study is actually from Harvard study, where they published this in Nature and showed that the subcellular architecture organization gets altered in disease, right? But also in normal, it has this original organization. And the same thing in the matrix or outside the cell, right? The matrix architecture, matritecture also changes. For instance, if you look at this blue-yellow regions, in the disease, it shows a bubble, right? While in the healthy region, it shows nice features of the extracellular matrix supporting the cell function. So therefore, it actually controls the cell and tissues. And to be able to understand this architecture, there are molecular screening technologies, right? There are sequencing, mass spec technologies, and many others. When you think about sequencing technology, you start with these cells, the architecture of the cells right in the tissue. But the problem is that we then dissociate this architecture, right? We basically use the lysine buffers to dissociate everything, and then you extract the DNA, RNA molecules and do the sequencing. But this is a problem when you want to understand the architecture. So therefore, we perform in our vision an image-based sequencing technology, which we call in situ sequencing, or now people call it spatial sequencing, that can help us sequence the cells before dissociating them. Basically, we keep the cell intact. We do not dissociate them. We keep their original positions in the cells and tissues. Then we perform multiple barcoding strategies to be able to read DNA, RNA molecules, as well as we will overlay them also with metabolites. Then we use typically microscopes or advanced imaging systems to be able to look at these molecular subtypes, then we will use the big data computers, right, or supercomputers to be able to convert these data sets to sequences. So this is, for instance, advantageous. Why? For instance, if you just do a single-cell RNA sequencing experiment, you get this soup of the cells, right? And this is actually similar to a fruit smoothie, right? If you start with the bunch of fruits, then put it in a grinder, right? Then you're gonna make a smoothie. And that smoothie then loses the original context of that fruit bowl, right? So similarly, single-cell RNA-seq also loses that spatial context. You can also only tell how many of these cells are there, and possibly also what are their types in the smoothie. But in the case of spatial or image-based technologies, for instance, this purple one is the H&E, or the histological image that typically pathologists would use, right, in the hospital. Then you're looking at different parts of these tissues to understand the health and disease. And again, this is the fruit bowl itself, and you do not dissociate this. You keep this intact, right? You keep this intact, and hence you can tell the different structures of this. So, but this is a spatial technology, right, which is called spatial transcriptomics, originally invented in Sweden and now commercialized by a company in the US called 10x Genomics. And here, then you're looking at the spots that are being measured, right, for across the tissue samples. But these spot sizes are around 50 to 100 micrometer. And if you remember the average cell size, that is about 10 micrometer. So each spot size basically would cover a very large area. Here, for instance, a spot size would cover this multiple cell area, right? But it doesn't necessarily tell you the single cell information about molecular densities across these tissues, right? So this is spatial technology. It still gives you the architecture, but one problem is that it averages out still a few cells, not millions of cells, but at least a few cells. Then there are third directions, right, around this, which can keep both spatial and single cell. And again, you start keeping the cells intact. You do not dissociate it, but you keep hybridizing labels and also fluorophores, the light emitting molecules, such that you can create these barcodes. And these barcodes then will give you this picture. And here in this picture, you're looking at each circle with different colors, and each circle, especially blue, indicates nuclear structures. And the different colors indicate, and particularly different distributions of RNA molecules inside these tissues. So therefore, these technologies can both provide single cell information, because you can see these single bubbles here, single circles, but also the colors indicate different cell types and different functions, right? So this is a timeline of this field. If you Google spatialomics, you will see there are a lot of materials coming up nowadays because it's a very hot topic. And why? Because after 2013, there was a boom and there was an interest to be able to measure more RNA molecules, more protein molecules, and also see the spatial information about how tissue looks, right, and how the architecture looks. And remember the first architecture that I mentioned? So that architecture is very key when you study the tissue, because the cancer, immune cells, and how they basically interact also is coordinated by the architecture of these tissues, right? So my entrance in this field is actually around 2014 when I was doing my postdoc at Caltech, where we invented the sequential FISH technology. It is a technique that can measure RNA molecules at very high resolution, at even super resolution levels. Also, it can measure the spatial information at in the tissues. Then around 2018 or so, I was at Stanford doing my instructor job, junior faculty job, where we also developed the Codex technology to be able to measure proteins and multiple of these proteins across the images, right? You can look at the previous image that I showed at the single cell level, look at about 30, 40 proteins all at once. So now the field is, of course, progressing indeed fast. And if you look at major journals, spatial, single cell, right? And all these titles are always coming up in the recent journals. So therefore, it's very important to consider to study the cell and tissue architecture, the spatialomics technologies, they basically landed themselves to these problems. And my lab specifically we are interested in not only the cell types, but also function, right? So when cells are in these tissue and cell architectures, right, or special tissue architectures, they are changing their identities, right? They could be the kidney cell, and they could be the epithelial part of that kidney or another immune part of that kidney. But while they're changing their decisions, they're always using these networks of molecules. So what are these molecules? For instance, the blue one is signaling, the black one is metabolic, and especially this red one and these networks are gene regulatory RNA level interactions. But if you can see, even in the subcellular level, the architecture matters, right? So there will be the molecular organization that can help us understand the interactions of molecules, and then finally it leads to decisions, right? And the cells either grow, right? They differentiate, or they commit to apoptosis and things like that, right? So therefore, we are interested in looking at signaling and metabolism along with the RNA networks, right, to be able to understand the cell function. So particularly, we are excited about mainly 3 directions. One is gene neighborhood networks, how to model high-dimensional data for interactive and also link to the patient outcome. For instance, here, you're looking at one single image, and inside of it, you have all these different smaller regions colored by individual groups, right? And this is not actually a US map, right? This is actually a single cell. But here, each group indicates that there are RNA molecules that are concentrated around those regions, right? So therefore, we will be talking about a technology that can model these interactions or neighborhoods. We call these neighborhoods, right? Architecture refers to the houses, apartments, and in the apartments, you have also your neighbors, right? You have one neighbor, another neighbor, and between you are basically talking and interactions with your neighbor, you decide on several things in your environment. So therefore, we're also looking at these neighbors among the RNA molecules, right, to also help us understand some of the cellular functions. Then we're excited also to study subcellular spatial signaling networks, where here also you're looking at these multiple colors indicating the signaling molecules at the subcellular level, and we are visualizing these networks of more proteins, right? And then our ultimate goal is to be able to trace the signaling transduction visually in the cell. Then we're also excited to study single-cell spatial metabolomics, particularly to understand how cells alter their metabolism, and also they follow a trajectory of metabolic pathways, right? They, for instance, choose the way that the glucose is used more, or fatty acids or fats are used more, right? Then we are also projecting these trajectories on the architecture maps, right, or tissue architecture maps as shown in here. So the first section that I'm going to talk about today is about neighboring RNA, right? So until now, people use these kind of technologies on the left, right? We had technologies to measure RNA molecules in the cell. Again, this is a single cell, and you're looking at these distinct colors. They correspond to different RNA molecules, and each dot corresponds to a single RNA molecule, right? Then it's dispersed across the cell, especially in the cytosol, right? And as you know, the RNA processing mostly occurs, especially translation process, mostly occurs in the cytosol, and you see this very high density. And this is pretty much common, but what's not common is that how do we look at these RNA molecules, and how do we also look at more than hundreds to thousands of different RNA molecules all at once by these images. And we generate these images by a technology called fluorescent in situ hybridization. But now using multiplexing strategies, we can then generate the maps of RNA for about hundreds to thousands of them. And then what's also common was that you can count number of RNA dots, as you see on the bottom left, right, across a single cell and say, oh, actually my blue dots here, we have about 2,000 of them, right? So therefore, actually this is currently about 1,000 dots. My RNA gene expression is about 1,000, right? Then if you zoom into other colors, I mean, the other colors are a little bit, of course, covered by the blue ones, but for instance, black one is also higher, right? So then you can look at the other black ones and other colors, right? And look at the gene expression by just counting these number of dots, right? Again, counts of dots will give you the gene expression. This is the akin to actually a single-cell RNA-seq expressions for the FPKM values. But in the case of these experiments, you're looking at just the counts. But beyond the counts, actually, we have another information that, for instance, the red RNA molecules are in the cytosol, but blue ones are in the nucleus. I mean, why are they in the nucleus? Because these are, for instance, non-coding RNA molecules called link RNA, and this is particularly MALAT gene. So therefore, it is operating in the most nucleus, right? It is different than the mature genes. But the interesting part is that the blue and red genes, they are non-neighboring, right? They're non-neighboring. Then, alternatively, you could also have genes like the bottom two, where the blue and red are neighboring each other, right? Neighboring each other. So how do you distinguish these two? Then we developed these computational frameworks, again, to cluster the individual RNA molecule positions, but ignore their RNA copy numbers, right? Ignore their intensities, ignore everything, but just keep their positions. That we have the X value, Y value, and if this is also in 3D, you have the Z value. So then cluster those positions using Leiden, and then it tells you basically multiple clusters, right? Let's say you can generate up to 30 clusters. Then you can zoom into one region here, a small region here, and inside that region, then you could study RNA neighborhoods by using the nearest neighbor algorithm, right? For instance, here blues are interacting or neighboring with each other, but here the blues and reds are interacting and neighboring each other. So therefore, you can distinguish these two cases. And again, these kind of technologies are generated by multiplexing these in situ hybridization methods. So for instance, if you have this RNA molecule here, this RNA is trapped or it's fixed inside the cell, right? There are a lot of proteins. You add formaldehyde, it basically creates this mesh of networks, and proteins are all trapped, and inside and near proteins, then the RNA molecules are trapped. So then you can add these red and green molecules and hybridize and image this under the microscope, and you see this dot on the cell image. Then you can remove this label by an enzyme called DNase-1, and DNase-1 removes all the molecules here except the RNA, right? RNA will be left intact or here. It won't be digested, but the rest of the molecules, they're digested, and then they're floating away the buffer, right? Inside the buffer. So then you can relabel the same RNA with a different color, right? And then generate a barcode for this RNA. For instance, you could color this with first green, then blue, and create this barcode of green, blue for this RNA. Then you can also perform an additional experiment to stain proteins using the antibodies that are tagged by fluorophores, and that tells you especially different subcellular architectures and how they co-localize with all these proteins, all these RNA molecules. Then this experiment you can do about 20 different cycles, right? And each hybridization removal and hybridization removal is defined as a cycle. Then you can perform the clustering, as just we mentioned, digitally, right, to perform the analysis that I mentioned before. So when you zoom in to a cell, then after the clustering the positions of these RNA molecules, you would obtain these boundaries of different regions, the region 1, 2, 3, 4, 5, 6. And we call these for now, and we define these as patch. And these patches, it will be each row, right, will be patches, and each column will be the numbers of these genes, right? So you're looking at the gene 1, which is the orange color, and that would be the number of genes within this patch. But instead of just looking at number of genes per patch, we're also looking at the neighborhoods that I mentioned. So you're creating this network of genes when they are neighboring each other. So we then call this technology SPA-GNN, Absolute Spatial Resolved Gene Neighborhood Networks. And this technology is unique because it has the properties in comparison to other technologies that can help us understand unique features, right? For instance, if you use co-expression, transcription factor binding analysis, computational net inference, or if you just look at some of these large spatial spot data, right, you could make all these networks of gene-gene interactions. But they don't necessarily immediately tell you the RNA neighborhoods, right? Because they are predictions typically. Either if the two genes are high, then you expect that they are basically networks. But in our case, it's a physical analysis, right? We call when the two RNA molecules are nearby or their neighbors, then they are the networks. So therefore, they are different than the other technologies. So for instance, we tested this in one of the data sets called MERFISH, originally invented at Harvard, but we also used this data set to demonstrate the idea. And for the same cell, you're looking at about 130 genes colored, right, in different part of the cells, right? So this is the same cell again over and over, but on the right panel, we just highlighted different gene subsets, right, just to show how it looks. Then you can create these heat maps of correlations across patches. You take the cell, chop it into small patches, then you correlate the RNA molecule densities across these patches, and this is what you get. And here, basically, you can at least understand the enrichments of RNA across different patches. Then you could look at the neighboring RNA molecules, which gives you high correlation value across the patches. But if the molecules, then red and blue, they are not nearby or they are not neighboring, then these patches will give you low correlation scores, right? So that's why you can use this metric to quantify non-neighboring. Then you can again start with the cell, zoom it in, and create these nearest neighborhood networks. So basically, you start with a central cell RNA molecule here. Then this RNA molecule, you find the nearest 10 neighborhoods. The nearest 10 neighborhoods, you count this, and you repeat this process for every single RNA molecule until you create this large network, right? And when you're creating this large network, so this is the large network, you start with looking at 10 nearest neighborhoods, then you do 1,000 permutations, then we perform z-score analysis of these permutations, especially for RNA neighborhoods, and that tells us the map of small network here or large network, and blue shows that they are not neighboring, and red shows that they are neighboring, right? Then these z-scores are also illustrated for this orange part of, for this purple part of the cell. And here, different RNA molecules, they are giving you different scores, right, based on how close they are, right? For instance, this blue and MALAT and THB-S1, they are not close to each other. Therefore, your z-score is very low. But for instance, these top two, the IG, F2R, and FBN2, they are near each other. That's why they are giving you high scores. Then we also tested the same analysis by GNN in another cell line, right, for the same technology, but this is a different cell line. Now this cell looks different, but we are getting the same different correlations here, right, for the second cell line, but the concept technically works, right, in even different cell line. Then when you look at two cell lines, right, and compare them for the same genes, right, fibroblast is one cell type and U2OS is another cell type, then you could see similar RNA neighborhood patterns, or you could see different RNA neighborhood patterns. So even if that you select two genes that are the same genes, across cell types, you might see similarities or differences, right? Then you could also compare these across many cells and generate all these patch correlations, right? And they compare them across the cell types. So which means that SPA-GNN, this technology can tell you, this algorithm can tell you RNA neighborhood differences across different cell types, even if you're looking at the same RNA. Alternatively, if you again look at the two different regions of the fibroblast versus the U2OS cells, and you pick three different cells here, right? And then you look at their nearest neighborhoods of RNA molecules, then you may obtain distinct RNA neighborhoods. Some of them might be similar, but in this case, the blue, which is fibroblast, it has a lot of more proximities or the neighborhoods of RNA compared to the U2OS cells. This suggests that there's a cell type specific RNA neighborhood is going on. Then beyond just looking at these images, we combine this across, for instance, 200, 300 cells. Then each dot corresponds to a single cell here. Then we classify these cells on a t-SNE or high dimensional reduction map, right? If you just use single cell RNA count and look at two cell types that I showed, then they're decently separated. I mean, they're close to separation, but they're also a continuum. Or if you use patch correlation, then still they're closer. But if you use the network or these patches or different regions of the cell, if you use those and then look at their variability, then you can perfectly separate this. For instance, by looking at the confusion metrics, you expect these diagonals to be high and other opposite black diagonals to be low. And the network variability gave us the perfect separation of cell types, right? The fibroblast versus U2OS, because it's the most sensitive one for the cell type separation. Then you could use less number of genes and perform the perform this analysis, and still you will have the same similarity of the clusters with 0.9 RAND score, which suggests that this analysis can also work with less and less number of genes. You may not always need hundreds of genes. Then you could also compare 100 genes and 50 genes and define these networks and look at specifically the neighborhoods. And neighborhoods are preserved across the subset of RNA-RNA pairs, right? So that also suggests that number of genes doesn't necessarily change the RNA-RNA neighborhoods. Then, alternatively, we did this technology. I think there was an image it didn't show here for some reason, but we did this technology, right? We used this technology to compare stem cells used for regenerative medicine treatment. So the first two types are human umbilical cord mesenchymal stem cells. The second one is human bone marrow mesenchymal stem cells. And the third one is human chondrocytes, right? So this is actually an output of the mesenchymal stem cell differentiation. But we wanted to compare these two cell types to the third one. And essentially also we wanted to see the differences of HUC and HBM. Even if they are mesenchymal stem cells, still we expect them to show some differences. So when we perform this, again, analysis of single cell into multiple patches, and you have the cluster numbers, then we have the correlations across these, right? And we then zoom into different regions of these stem cells and perform the nearest neighbors analysis, and you'll be obtaining these networks of RNA molecules, right? Then in the same cell, for instance, for this cell, different part of the cell shows a different RNA neighborhood network. And another part of this cell also shows the second cell type shows different RNA neighborhood networks. Again, not only single cell now varies, but different parts of subcellular regions also vary. And then finally, this third cell type, right, and different color regions, right, the green region versus the pink region, we all have different RNA neighborhood networks. So, but using this kind of, again, network variability concept, by using the RNA count or expression. So this is how you do normally cell classification. You just use the RNA count and you classify genes, or you compare the correlation across these small patches, or you look at the network variability. So what we found out is that actually you can define cell type separation better, right? With this is only count, this is only patch, and the third one is the network. Again, you want this colorful diagonal to be high and black diagonals to be low. And we got the lowest black diagonal numbers in the confusion metrics, right, to achieve a better separation of cell types. So this suggests that actually the network neighbor variability approach can be a good way to separate the cell types. Then you could also truncate, look the same cell into a smaller cell, but just truncate from the edges, and you'll obtain still similar patch architecture. Then we could also look at the same cell, another cell type, right, the stem cell, for instance, and measure the genes responsible for cytokines. And we every now and then observe some of the genes are actually localized at the edge of the cell. Then you could use a different geometry of patches. Remember the previous geometry, it was a US map almost, but now you're looking at the almost geographical map of the latitudes and altitudes almost inside the cell. Then you could also define these RNA neighborhoods across different parts of these cell. Then using this information, you could compare the cells that have edge expression versus non-edge expression. For instance, this cell on the left shows the edge expression, but on the right, you're looking at that these edge expression ones are only the pink ones, or this color, bright color, but blue ones are the non-edge expression ones. Then you could use this concept to combine RNA and protein neighborhoods. So for instance, here, collagen 1 is a gene encoding for collagen molecule, most abundant molecule in the human body. And these are the green molecules. So then the CON-A and WG are protein stains for endoplasmic reticulum and Golgi molecules. So we know that actually collagen is associated with endoplasmic reticulum, right? Just because it is playing an important role when the collagen is secreted, right? The ER basically regulates this process. Then we further, again, did this a patch map, the US map type of map inside the cell. Then we created these networks, and we showed that when you look at correlations and z scores, particularly the blue, which is that the collagen RNA molecules are enriched on ER molecules, but not Golgi, right? Blue tells us that the collagen is enriched on ER. Again, it tells you that the collagen is the neighbors of endoplasmic reticulum, but not Golgi molecule. So this was the story about how RNA neighborhoods can be measured in a subcellular architecture, right? So next, I'd like to switch gears to signaling biology to understand how signaling molecules, which are proteins, right, are organized in the cell. And we are studying signaling because for especially lung cancers, unfortunately, existing chemotherapy approaches, they can only extend the lifetime by 5 to 11 months. But the most recent drugs, which is called osimertinib, those are tyrosine kinase inhibitors, so they can extend the lifetime by about 20 months. So, but still, even if they extend the life by 20 months, some patients, the red ones especially, they become resistant, unfortunately, and we lose our loud ones just because of these. And we think what's happening in these kind of red patients is that the signaling pathways are so complex that even if you block this EGFR, right, this molecule, right, this is a signaling pathway, still the other pathways, they regulate the process and then they're leaky. They basically bypass the drug as well as the effect of the drug. Then they lead to finally the growth of tumor, right? They don't get basically suppressed. So therefore, our goal is to be able to use this technology, implement it on a microscope using microfluidics. We perform antibody stains of protein, and then we repeat this multiple process. And this is how it shows multiple staining and labeling. Basically, we are now using robots to perform our experiments, right? Of course, students and researchers are great in pipetting, but we are trying to reduce these times, right? And then finally, we are generating these kind of setups where you have the tubes, the autosamplers, and then the microscope has this stage that has the multiple fluidic inputs and outputs. And you just basically take the sample, put it on a microscope, and study these cells. And this is a technology that we use to understand signaling resistance. And when you compare it to the other technologies, the red ones actually are important. We are cutting down the experimental time to only a few hours per cycle. That allows us to generate rapid profiles. And specifically, what we want to do is to localize protein molecules in the cell by images, then use these images to define signaling networks. Until now, what people did is that they took the cell, they used an antibody molecule, they targeted all these proteins, then they pulled down all these proteins and looked at which protein was bound to which protein, right? But now what we want to do is that don't dissociate the cell, just keep the cell as is, then tag protein molecules and measure the distances and their interactions in the cell. And this is how we do these things, right? So for instance, each rectangle here shows 3 cells. But this is, of course, among a large plate of experiment where you have like thousands of cells. But then these 3 cells show epigenetic markers in the nucleus, and the signaling markers are in the cytosol, right? I guess can be seen in these images. And then the inside of the cells, you see a dark hole here because the proteins for signaling typically are enriched in the cytosol, but not in the nucleus, right? So therefore, you see these holes. But then interestingly, you can do 2 things. One is you can identify the nuclear region and cytosol region, and the red here is the nucleus. Then this type of an analysis allows us to study the translocation of protein molecules between nucleus and cytosol, right? And hence you can understand the signaling dynamics. Then we perform correlation or intensity analysis across multiple single cells and their pixels. And for instance, the 2 signaling molecules, they show correlation in these multiple single cells. Then we perform data science with this approach. For instance, take all these 30 markers, multiple protein molecules, put it on a single map of UMAP, right? So this is a UMAP. It's a high dimension reduction. Then color them using light and coloring. These colors then are projected back to the cell image. So this outside boundary is the cytosol of the cell. Inside boundary is the nucleus of this cell. Then the colors indicate the colocalization of subset of signaling molecules, right? If you have AB molecules, it will appear here. If you have CD and EF molecules, right, then they will show different groups. So this suggests basically the cell has this very complex signaling organization. Again, back to the architecture, signaling molecules even use different part of the cell to perform different tasks, right? It's actually important to compartmentalize even signaling molecules. Then how these architecture of the subcellular regions look, right? you could see all these individual single cells. They are totally different, right? So you have basically one apartment complex, you have another apartment complex, and their architecture is different, right? So then you could see exactly the same thing here. All these individual cells, they have different signaling architecture, right? And when you zoom onto the same colony even, right? Let's say you have one apartment complex, and inside each apartment, of course, you have a different organization of the intra-apartment, for instance, where the chair is, where the TV is, right? So then you're looking at all these protein molecules playing a function in different part of the cell. For instance, this green part that I indicated before was with AKD molecule, right? The other part was PAKT. And this red part is again, epigenetic molecules, right? Here, these are the epigenetic. And PMTOR is also on this side and things like that. So basically, it shows us how signaling organization can be different in different architectures of these cells. And here's the three-dimensional visualizations of this, where you're looking at the brown as the nucleus, and outside boundary is a single cell. And then the other colors show the signaling colocalizations with subset. For instance, this blue one is different from one cell to another, and it suggests that the single cells are heterogeneous, and that's also important to control signaling resistance. And to study our initial hypothesis that how signaling, spatial organization of signaling molecules are altered by disease, and then how that leaky signaling pathway can give rise to resistance, right? We then tried this direct treatment of osimertinib, and this is used in clinics. We compared the direct treated with untreated one to see how it affects our cells. First, when we perform the multiplex experiment, we start to see that the PAKT pathways, they get suppressed, and then PEGFR start to get more discontinuous, which means that they are blocked on the cell surface. But then the Wnt start to become more higher expressed, just because that's a leaky pathway in the cell. So Wnt resistance is known in lung cancers, but we also observed this at the cellular level. Then we compared the untreated and treated cells, and PEGFR and PAKT were the markers suggesting that the drug worked. Then we also compared cells that are treated with control and all the way up to 60 nanomolar concentration of drug. And the more we treated the cell, the more the PEGFR got more discontinuous. What does it mean by discontinuous? Do you see how these patchy green here, those dot, dot, dot structures, they are discontinuous. But in the original one, PEGFR were more continuous. So everywhere you had a PEGFR. So this suggests that physically, the molecules are bound, and then they are basically inhibiting your molecules. So then we compared no drug with drug at the single cell level, and the blue ones are with drug, and red areas are without drug, and each row is a single signaling molecule. These are proteins. We were able to also recapitulate the stratification of these. Then we did the same high-dimensional clustering of pixels in the single cells. So here, these are the, again, cluster colors coming from 30 multiplex markers. And in the control versus drug-treated one, we observe clear differences, right? How do we visualize it? We will use network visualizations. For instance, here, each bubble shows the percentage of cluster of all clusters. And then the pixel-to-pixel closeness will be the blue lines. So they're basically spatial proximity. If you have a blue pixel, right, and a yellow pixel, then between them, if they're very long, then you measure this in the image, then you project it onto here, and then it will be longer, right? So then in the control versus direct-treated one, what we observed was that direct-treated one started to break these distances across into smaller distances, right? And then these bubbles start to get bigger. So for instance, 2 and 6, in the previous one, it was smaller, and this one was high. After drug treatment, it becomes higher. This suggests that the Wnt pathway is leaky, even if there is a drug treatment occurring. And for instance, the 14 here is getting smaller. This suggests that the drug treatment is working. But Wnt pathway is suggesting that that's becoming more prevalent among the clusters of your cells. So then the 6 and 2, they're getting bigger in terms of the dot size, right? So that's important to distinguish. Then we did this also in patient tissue. So here's the tissue architecture again. The yellow is cancer, and the green is non-cancer. We just focused on the cancer regions and showed how signaling molecules are distributed differently across different regions of this tumor. Then we compare these across 21 patients, and for each patient, we have 3 different replicates. Then when you zoom in, it's basically a heterogeneous expression across tumors. And particularly this bright area corresponds to tumor area, dark area corresponds to non-tumor area, which means that tumor upregulates signaling, but the level of upregulation is unique to each patient, right? It is heterogeneously expressed in each patient. Then in the last part of my talk, I'd like to highlight the metabolism and cancers. So as you know, metabolism is altered in cancers. Particularly, it is increased in cancer cells, and immune cells also, they reprogram their immune metabolism. And also our life also alters our metabolism, right? Depending on how we exercise, how we eat, fat diet, or we are obese or not, right? Or do we sit every day, or we stand up every day, right? So this metabolism alters. So we suggest that actually we should think about cell and tissue architecture from the point of view of how we can incorporate the lifestyle and metabolic measurements such that you prescribe drugs, right, for especially cancer patients, considering both. And then you might be, and you might end up getting better treatments, right? Because we know a lot about us and the tumor molecular profiles, and then we can treat them better. But the problem is that all these metabolic assays are also like making smoothie. They're not giving you the individual single cells. So we think that we can study single cell metabolism across tumors, right? And also we can compare metabolism across patients. And when you zoom into a tumor, that's what you see. The normal tissue will be on the left. And inside of the tumor, you see all these multiple interactions of cells that some of them actually, they compete with the molecules, right? They actually, for instance, secrete lactate, then they change the immune profile, or they compete for the glucose inside the tumor region. So there's a lot of competition and interaction going on. And we developed a technology for this where we started with H&E imaging, or the histological imaging of this tissue, annotated different regions. Then we stained another separate tissue with these antibodies that are tagged by not fluorophores, but something called isotopes. We are using isotopes because we can look at 35, 40 molecules all at once. Then we perform one imaging using a secondary IMMS spectrometry. It says 3DSMF, this is our unique name, but it's a metabolic imaging technology. Then we perform protein imaging, then we overlay these two. And in the imaging, we perform processing and clustering and data analysis. So this is a crowded slide, but you're gonna see in the next slides. So first, in the tumors, we can use protein staining using isotopes and on the antibodies, right? Remember isotopes. Then you can look at all these proteins for cancers, stromal cells, macrophages, and T cells. Then when you zoom onto different regions, you can color these cells by their identity. Then you could then create these graph networks right at the end. So each dot corresponds to a single cell type. Then we can look at their interactions in the different parts of the cell. Then you start with this again, isotope and antibody complexes, right? You can use 30 of them and stain your tissue, then perform metabolomic imaging, protein imaging, and then combine these two data sets. And zooming into one lung cancer patient biopsy, here the orange color shows the phosphate molecule found in your DNA, but there's no label. This is label-free. Then spatial metabolomics shows you all the metabolites distributed across your basic nucleus region. Then nuclear metabolites that are tagged by antibodies targeting the histone antibodies inside the nucleus. Then also these right colors are the surface receptor antibodies that are targeting the CD8, CD68, collagen, and Ki67, and all that at the cellular level. So it tells you the cell type. So then one can quantify the cell types from the protein information, then overlay with metabolic information. That's what we did. At the single cell level, we got the lung cancer patient samples on a microarray. Then we performed proteomic classification. Right here, the C shows how the bright colors are the immune cells and the cyan color is the cancer. Then we grouped this as tumor, stroma, and others, and showed that the tumor has a higher expression of several lipids, glycine, compared to stroma or non-tumor areas. Then we could also go back to the original image and color the metabolism of lipids, glycines, and cholesterol at the single cell level. So this is actually very exciting for us because for the first time, we can do all these kind of studies for hundreds of metabolites at the single cell level. Then the data science allows us to compare the profiles of about 23,000 cells for 40 metabolites. And bottom, you have these 20 different proteins. Then you could use the protein for cell types and metabolites for metabolic analysis. And further, you can subgroup the metabolites into glucose-associated, cholesterol, and amino acid associated, right? As well as the lipids. So then several tumor regions have higher expression of lipids, but several others are higher in the stroma regions, which suggests that lipids are depleted or generated in tumors. And it's basically time-dependent and energy-associated process. Then we can further associate the tumor immune stroma, separations in the tumor. Right here, you're looking at 3 different colors in the tumor region, right? And then by counting number of immune cells and their metabolism, you can create also these immunometabolism maps. Immunometabolism is the metabolism of immune cells inside cancers, right? That's what you're looking at. So then we can also compare the metabolic profiles across patients. But one thing that I want to highlight is that because this is a multimodal data, we have metabolites and proteins. You have the 2 different UMAP or the high dimensional representations here. If you were to use the existing data integration approaches, so this is about bioinformatics problem, right? You can look at the one data, it shows all these colors. The second data shows similar colors, but there are a lot of shared features. But what we found out is that in our case, the metabolites and proteins are 2 different worlds, because proteins can tell you the cell types, but metabolites, if you have a lipid on the cell surface or cell membrane, that lipid may be every cell type, right? So you cannot basically distinguish them. So therefore, my student actually developed this variational autoencoder approach that combines 200 metabolites with 20 proteins. Then this is integrated in the latent space of this variational autoencoder. Then we create the cell type specific maps of metabolites, right, in multiple patients. Well, what did we learn? We learned that the metabolism and protein expressions are unique in different patients. Then several metabolites are lowly varying. For instance, each row here is a different patient, and then they are lowly varying. But also there are some other metabolites that are highly varying, right? So the blue and then orange one, they're highly varying. Then, for instance, when you use the t-SNE after the data integration, then each color shows the different regions for the same patient. For instance, orange, they're closer to each other. These are from the same patients. And for instance, here the purple, they're from the same patients, but different regions. So it shows that the metabolic changes of cell types are patient-specific, but also they can be different from one and another, right? So it's a very complex regulation. So we think that actually now you can combine immunotherapy with metabolite supplements to increase the survival. And what we propose is that now use the lifestyle, diet, and BMI for designing patient therapies, right, especially for immunotherapy. And you could also think about simple diet prescriptions to boost the immune system, especially for immunotherapy responses, right, how they increase them. So there is a good in the Cell paper, right, demonstrating the initial bits of it, but that's what we are moving after. We have now the technology that can measure single cell metabolism and proteins. And now we'd like to also use metabolites on top of immunotherapy to be able to increase the responses to these lung cancer treatments. And now I will be summarizing. So we have shown today how we could do graph-based images of proteins and metabolites, right, where you can look at single cell differences. We also talked about spatial signaling networks and how these distinct colors show the protein molecules and their networks. Then in the first part also, we talked about RNA neighborhood networks, where we also highlighted how distinct part of the cells show different networks. And I'd like to also highlight that the virtual reality can be also integrated into such an exciting data where you can discover the patterns, right, of neighborhoods, right, RNA neighborhoods, or protein-protein interactions, right? And all these at the cell using virtual reality. So this was published in Cell Reports Medicine. You might want to read this. It's a fun article showing how biomedical field can use virtual reality. Again, even for bioinformatics community, this could be enabler, right? It's an interactive technology. So with this, I'd like to wrap up, and I'd like to thank to our collaborators from Emory University and Georgia Tech. And I'd like to gratefully acknowledge the support of generous funding agencies. And of course, without the hard work of the team behind such a work, right, this would have not been possible. So since from 2019, when I started my lab, now we're growing, we are branching out to multiple directions, as well as collaborating with large consortiums like this one. And with this, I'd like to conclude, and thank you again, Bioinfocongress for this kind of invitation. And I'm happy to take any questions if you have. Thank you.
Thank you very much for this precious presentation, Dr. Coşkun. Then I'm starting with the first question. Why are preclinical models inadequate, especially in tumors? What are the challenges in developing prototypes that mimic the tumor microenvironment? Oh,
yes, I think this is a great question. We just actually published with a colleague of mine a paper, a model to mimic tumor microenvironment, right? So this microenvironment is typically framed as organoid. They are mini organs, and you basically mix cell types with the extracellular matrix compartments in a dish, right? You take the matrix with cell types, you mix them in a dish, and then you let them self-assemble, grow, and make it a synthetic tumor microenvironment, right? In our last work, which was published in Nature Materials, that's what we showed, that the lymphoma organoids can be recapitulated by using the chemistry that's mimicking our body's collagen chemistry, right? And then you mix with the cell types, and then they grow and give rise to the functional signaling and, for instance, metabolism, right? Profiles of cells in the organoid platform. So it's actually combining our also spatial omics technologies, right? Because important that once you make the synthetic environment, you want to also understand how they're similar to the real tumor microenvironments, right? You could actually look at their expressions like high-low, as well as their spatial architectures, right? Are they preserving the cell neighborhoods, right? Are they preserving the original architecture, right? So that's why we are currently actually developing experimental as well as computational frameworks to establish this, right? Synthetic tumor microenvironments benchmarked by the architecture concepts, right? And then use iteratively, right? Use chemistry and bioengineering to make additional, new synthetic tumor microenvironments. But that's a very important one, just because also right now there's a trend in the US and in internationally also that you want to decrease the animal use in your research. Can you use an in vitro platform like an organoid to be able to study the disease response, right? And then at the end of the research, you should be able to replace the animal studies with just a bunch of microwells, right, or microfluidic chips, right? Then you can actually get the answers of which drugs will be more efficient, which patient will most likely to respond to which subset of drug, right? All these kind of responses. Therefore, I think your question is really important for us to impact the future of medicine by reducing the animal use, right, but increasing the clinical relevance of synthetic tumor microenvironments.
I appreciate your answer, sir. This is from one of our YouTube participants. And next question is, a current challenge in two-dimensional analysis of tissue is the detection of structure independent of the cutting angle. How can we get robust morphometry of tissue structures?
Thank you, Ayşenur Kaya, I think, for this great question. So I think what Ayşenur is specifically referring to is that when you look at the tissues, right, there are a lot of features. Some of them are sharp, like their edges. Some of them are smooth, they are more continuous. So identifying the features in this tissue, along with, of course, expression of many markers, is a challenge in image processing. Actually, I'm teaching the medical image processing in my institute, so therefore we teach all these filtering and other methods, right, to perform edge detection and beyond. But now classical edge detection algorithms, classical image processing algorithms are lacking behind. So what we are doing in my lab actually is to be able to identify these for instance, graph-based architectures, right? You make basically individual dots in your tissue, right, as a graph. Then you take that graph and then project it in a machine learning, deep learning interface. Then that interface, as you compare multiple tissues and multiple different tissue regions, is much more robust, right? So you might want to check, or I can refer to new articles studying the morphology using this kind of approaches. For instance, there's an algorithm called Vampire, developed by Johns Hopkins professor Dennis Wirtz, and they're defining the cell shape by getting the edge of the cell as a point structure, right? They basically take a cell and make it point, and then they take these points and their positions and project it on a PCA, or principal component analysis, high-dimensional, three-dimensional space. Then there, they can go back and say, oh, actually, my cell type is round. My cell type is elongated. My cell type is, like a square. I mean, nothing is a square cell, but my cell type is like square, right? So those kind of methods actually are much more robust. So therefore, I highly recommend considering integrating morphological analysis of tissue and tissue structures, right, by not just the filtered or image processing techniques, right, but also new machine learning algorithms to group them as the graph-based methods, right? That's what I would highly recommend. I think it's a very good question.
Thank you so much for your answer. The next question is, why are targeted therapies for neuroendocrine prostate cancer and the castration-resistant adenocarcinoma tumors not available? Could tissue architecture be modeled?
Thank you. Well, this is very specific. I'm surprised. So this refers back to our paper also with the same team that we published 2 years ago in Advanced Materials, where we also use the same chemistry, hydrogel chemistry, to mimic the prostate cancers, right? And then these are the NAPC, right, and CRPC adeno. So these are specific therapies that shows a different molecular profiles, and they are not necessarily responding to what's available as a treatment, right? So I think what the question is asking about, tissue architecture can be modeled. Yes, I think if you can model the prostate architecture, distinguishing the NAPCs, CRPCs, which in that and Advanced Materials paper 2021, we have the organoids made by our colleague, and we also looked at the NAPC and CRPC tissue microenvironment in also cancer patients, and we saw a lot of similarities, right? We saw a lot of similarities, but there were a lot of still differences. So currently we are optimizing this further. And as you mentioned, if the tissue architecture further mimics the exact NAPC and CRPC tumors, then at the end of the day, you'll be able to mimic this much easier and better, right? In terms of finding the better treatments for these unique prostate subtypes, prostate cancer subtypes. But thank you. I think this is a very nice question, suggesting that clinic has some unmet need, and bioengineering and bioinformatics can meet together, right, to answer these questions.
Thank you so much for the answer. Our next question is, how can we identify and quantify organ-specific microanatomical domains from biological images without previous knowledge?
Okay, great. I think without previous knowledge is key there. Thank you, Nagihan. So how can we identify quantum organ-specific microanatomical domains from biological images? So assume that, for instance, we collect tissue images from multiple organs, right? And let's say you do kidney, prostate, right, lung. Then you get all these H&E features, right, or the histological features, or even immunofluorescence-like features. Then you create these, maps of their cell stains, and then their anatomical features, right? You can look at, this is my epithelial region, this is my, other part of the, prostate, or this part of the kidney, right? So then using these three, what we would do is we would take the single cell information from this, basically color one cell at a time, right? One cell at a time. For instance, in the prostate, you have 100 epithelial cells. In the kidney, you have 200 epithelial cells. And in the prostate, you have another 300 epithelial cells. So there are multiple organs, basically. Then we would also take their distances between them, right? These 100 cells, they have very close distances. But in the kidney, these 200 cells, they have separated distances. They are distant from each other, right? Then by using this two information, you would try to create a comparative analysis using a latent space model, right? That we just discussed, the metabolite and protein data that I mentioned, right? One of them has more markers than the other. But then what you do is in the deep learning space, if you convert them to a latent variable, right? Latent variable, then that latent variable will be able to combine them all in a single map, right? Then you don't need any previous knowledge, right? You don't need any specific training. But all you do is when you do the latent space combination, then in that combination, you can actually then project them on a high dimensional visual or cluster that will tell you the organ-specific features on a heat map, right? It can tell you number of cells. It can tell you the distances in between them. But again, it's important to compare them in the latent space, but not originally. If you just take this data set, put it in a heat map, it will be difficult to combine because there are different cell types, there are different distances and all that, right? So therefore, you need to be able to distinguish the two. But this is a great question. I don't want to, of course, take too much of your time, but I would suggest you think about latent space combination for this.
Thank you for your answer. And next question is, in spatial, technologies now enable the characterization of heterogeneity of tissue architecture and the opposite of the novel data can detect spatial information. With this limited information, how could it be made clear the relationship between cell-to-cell interactions?
Yes, I think this is a good question. So I think this refers to the fact that most of the spatial technologies, they choose the genes to measure. Let's say you measure 100 genes, but the genome-wide information is 20,000 genes, right? But 20,000 genes RNA sequencing, for instance, can discover new information. So what we are doing currently is to integrate both worlds, right? By using, for instance, the information from 1,000 genes, you can define the cell-cell communication interactions, but then look at certain interactions of, let's say, subset of 20 genes. Those 20 genes, go back to the original discovery data, RNA-seq data, and find, similar to these 20 genes, what are the other genes doing similar functions, right? Then if you integrate these two data sets, then you can infer that, oh, these 20 other genes that I didn't measure in my spatial could also be related to my cell-cell interactions. And as a result, you can fill the gap in your information. I think the key is the hybrid analysis between the spatial and non-spatial data together. There are actually very nice works recently published by our colleagues I can refer to. If you're interested, just send me an email. I'll send you some of these recent publications. And that's what we were also doing in our case. If you look at, for instance, 100 genes in stem cells, and then look at the databases measuring 20 genes for stem cells, then can you actually find the differentiation of stem cells in the differentiation path, right? Much better than, right, just the spatial only. Because spatial is really limited in terms of number of genes, right? But then using the non-spatial data, you can find associations of those non-measured genes, and that can tell you this differentiation path much better than only looking at the spatial data.
I appreciate your answer, sir. Our next question coming from YouTube participants. Was clonal heterogeneity in samples evaluated in your SPA-GNN study? Did you decipher the genetic purity of MSC colonies? Thanks.
Thank you. This is a very interesting question, Bedish. And so in terms of clonal heterogeneity, so currently we have not compared this for MSC or mesenchymal stem cell colonies for how a genetic purity of a colony compared to another genetic with a different clone, right? How they compare with each other. I think that's our next step. And we would like to compare with, for instance, take an MSC and start mapping their colonies, right? You have this colony with this colonal architecture, another clonal, then fix the sample and continue, right? And then continue by using SPA-GNN. I think that's going to be our next step. And that will be very exciting because then you can look at how clonal information generates different subcellular networks. But thank you for the suggestion.
Thank you for your answer. And our last question is, Which advices Dr. Coşkun may give for students studying for scientific fields?
Thank you. I think you are doing very great work here with Bioinfocongress, integrating bioinformatics to research programs, big data analysis, machine learning, AI to biology is the future of medicine. I highly recommend students to learn how to code as early as possible, right, in their careers. And also, I highly recommend them to reach out and find networking opportunities within this community and do internships, right, summer studies, as well as pursue research opportunities as early as possible in these fields. And I hope in the near future, AI combined with biological, problems will really take over all the possible strategies that can help us solve the challenges of diseases, right, and many others. I think this is what I would recommend. And I think that you're doing really, again, an important key piece here, which is bringing the people internationally all into one online platform, right? And I highly recommend that your students here in this platform should reach out to these highly, interesting, research groups, then start working with them together, right? And that will, of course, increase the impact of our students' future studies.
Thank you so much for your advice.
Yeah, thank you for the kind invitation. And sorry, I think we're a few minutes late, but yeah, I'm happy to answer any questions if you may have separately on, offline. And my email should be online, or it should be also with you guys in the Bioinfocongress. They can get my email from you guys, and I'm happy to answer offline questions. And thank you again, everyone, for organizing this fantastic event.
On behalf of myself and all our participants, I would like to thank you for spending your valuable time, transferring your knowledge to us, and for this informative presentation, sir.
Thank you. Have a nice day.
I wish you healthy days.
Thank you. Teşekkürler. Bye-bye.